Good afternoon, everybody. This is Nelly Poyak from Dana Farber. Thank you for joining us for this seminar series. And just as a reminder, the talks will be uploaded on that website that you see on that link on the first slide. If you don't have time to stay or want to share with others, you're welcome and they can look at it later on. Also, we have questions that you can ask through the chat function, the Q&A on the Zoom, and we will answer some questions on line, depending on in live at the end of the talk, depending on how many we get, and then Len will have access to the questions later on, and he can answer those as well. So it's my pleasure to welcome today's speaker, Len Zahn, who's a Grusbeck Professor of Pediatric Medicine at Harvard Medical School, investigator of Harvard Hughes Medical Institute, and he's the director of the stem cell program at Boston Children's Hospital. And Len's talk today is entitled Stem Cell Clonality and the Niche. So thank you, Len, for doing this, and go ahead and start.
All right, great. Well, I want to thank Nelly and also David for setting up this amazing series. I have a lot of friends who are speaking in it, and it's quite a good group. I also want to say that I certainly wish I was doing this lecture live, and I'm very appreciative of the medical staff and of the patients that are going through a lot right now. So again, it's a pleasure to give this lecture. So just as disclosures, I'm a founder and on the scientific advisory board of Fate Therapeutics and Scholar Rock, Camp4, and Amagma. And the outline of my talk, I'm going to give an introduction to the stem cell niche. I'm going to define a role of endothelial cells that we believe is critical for function. I'm going to talk about some new methods to barcode hematopoietic stem cells and blood cells. Then talk about pathways that are regulating clonality, and then defining a role of macrophages, and then some concluding remarks. So I focused most of my career on studying the hematopoietic stem cells and the niche. So as you may know, there are inside your bones, you have your bone marrow, and that's where the stem cells reside. There's a constant influx and efflux of hematopoietic stem cells. These stem cells have the ability to self-renew, or they can differentiate into the blood lineages. Now, we know that this process works, and a patient can have a bone marrow transplant and rescue the immune system from lethal irradiation or chemotherapy. And that process is curative and only needs to be done once. But still, about 25% of patients will die from the transplant procedure or complications. And so there's a need to figure out other ways of helping the transplant process. Now, this process of self-renewal here is facilitated by the niche cells. This includes the vascular endothelial cells here, the stromal cells, and even the bone cells themselves. And so we wanted to dive in and understand the niche. And as many of you may know, my favorite organism is the zebrafish. And I just want to tell you about how blood stem cells are actually formed in the zebrafish. So here's a 36-hour embryo, and there's the heart and the dorsal aorta, and the stem cells are actually born in the aorta. So we're going to dive into the aorta, and what happens is a blood vessel, an endothelial cell, will transform its fate, and it will round up to become a blood stem cell. That cell could be transplanted at that point, but endogenously, it goes into circulation, and it's going to go round and around circulation, and eventually land in the site where blood cells will be formed next in the animal. In the fish, this ends up being in its tail. In a human, it would be in the fetal liver or subsequently in the bone marrow. This stem cell will then transit into the niche and will divide. It will then enter back into circulation and colonize the next niche. This is in the marrow. So again, in humans, this would be in the bone marrow. In the zebrafish, this is actually in the kidney. And this makes the kidney a hematopoietic organ. Some of these cells actually bypass the kidney and go right to the thymus. And this will start the immune system. And all of this allows this animal to make blood throughout its entire lifetime. So we've been studying this process, and we had the ability to make zebrafish that had green stem cells. We took an enhancer that is blood stem cell specific. It's a mouse RUNX1 enhancer, and we drove green fluorescent protein, and we mated this to a fish that had red blood vessels. So you can see up here at the top, the dorsal aorta has blood stem cells that are being born. They go round and around, and then they're going to land. Now, being a hematologist, I've done marrow transplants on patients, but unfortunately, we're all opaque, so I've never seen a stem cell land as it colonizes a hematopoietic organ. So if you could focus on this circle, what you'll be able to see is the stem cell comes in, transmigrates outside of the blood vessel, but then something strange happens. The blood vessels actually wrap right back around the stem cell in a process we call endothelial cuddling. This stem cell divides, 90-degree turn, and one stem cell leaves and the other one stays. And this is a very stereotypic behavior. So we wanted to understand more about this niche and the live niche that we're seeing. And we took this very animal and we did something called correlative electron microscopy. What we are able to do is solve the structure of the niche. So in this particular case, what you can see is the stem cell is surrounded by 5 endothelial cells. And work by Owen Tamplin, who has his own laboratory at University of Illinois, actually has been able to show that the exact same niche structure exists in the kidney marrow also. So this is, again, 5 endothelial cells. And surprisingly, this stem cell is actually physically attached to a stromal cell. And it turns out that this stem cell that's been going round and around the body, when it landed, it abutted against a stromal cell, and that's the surface on which the stem cell divides. So almost all divisions are seen in contact with the stromal cell. So this niche has many different cell types, and we wanted to understand it better. And so we elected to do a new technique called RNA tomography, or TomoSeq. What this is we orient an embryo kind of perfectly up and down, and then start sectioning the animal from the top of the animal, the dorsal, to the ventral side. Then each section is actually put in a tube and submitted for RNA-seq. Because the niche is in this ventral region here, we'll end up getting niche-specific genes. So this worked very well, and if you look at epidermal genes, you can see they're on the outside of the fish. You can see the spinal cord genes run exactly where they should. And what we saw was about 144 genes that were enriched for where the stem cells were. These are genes that are not in the stem cell, but they are expressed in the cells adjacent to the stem cells. Now, what we found was there were 29 genes that were enriched in those endothelial cells that are doing the cuddling reaction. And we knew this because we had previously sorted endothelial cells from the embryos, and a number of these genes shown over here were endothelial specific. But then we did in situ hybridization to show where these genes are found. And if you look at this one, KDRL, that's the FLIC1 receptor, and it's in all endothelial cells. You can see them here in these intersemitic sprouts. But then these 4 genes came out of our analysis, E-selectin and a mannose receptor, for instance. And when we do in situ hybridization, pretty much the only place where these genes are expressed is where the stem cells are. And you can see very specific expression in the niche. So what this represents is the vein that is in the marrow, the venous sinusoids that are specifically adjacent to the stem cells. And what we've defined is the sinusoidal program. So we were very interested in understanding this more. And we took the promoters of E-selectin and of the mannose receptor, and we drove green fluorescent protein. And what you can see here is that this 1.3 kilobase promoter is enough to drive expression just where the niche is. And that allows you to sort these cells out and be able to study them by single cell technology or by bulk analysis. What we've done here is to use a technique called ATAC-seq. ATAC-seq looks at how chromatin is open. And what we found were peaks that were open in the niche endothelial cells, but not anywhere else in the body. And we took those and coupled them to green fluorescent protein and injected them into the fish. And what we saw were the endothelial cells of the niche were lit up. We then did this with a number of other genes. Same thing, you can see very specific peaks here. And again, you can see the expression pattern is very, very specific. So we took 12 peaks and we studied them functionally for GFP expression. And we found that 12 out of the 15 peaks we picked were driving expression correctly. Now, there were some peaks that were present both in the in this region, the niche, but also in other endothelial cells. And when we drive expression of those, you can see multiple different patterns here of endothelial cells. So all endothelial cells light up. And this was 6 out of 6 ATAC peaks had this pattern. So this suggested that there was a code. There was a code for the niche endothelial cells, a transcriptional code, and we wanted to discover it. So one of the things that you can do is to look at the motifs of DNA motifs that are present underneath those peaks. And what we found is in the niche program, the endothelial program, you had an ET site, a SOC site, and a nuclear hormone site that were enriched. Whereas when we looked at the pan-endothelial peaks, they were mostly just enriched in the ETS motif. So we wondered, given that we have a little bit of a code here for driving endothelial expression, we looked at our RNA-seq, and there were 7 transcription factors that were expressed at a high enough level that could bind those motifs. And these would be possible drivers of that cell fate. So then what we decided to do was to take all of the niche factors and put them on a ubiquitous promoter and see if we could reprogram parts of the embryo to become an ectopic niche. So what you can see here is in the control, down here in the E-selectin, you get your normal pattern. But if you've injected all 7 factors, you start to get extra blood vessels. And using our transgenic line, you can even see those extra vessels there. So this shows that these factors have the ability to reprogram cells to make a venous sinusoid. So then what we did is we got this down. We did an N-1 experiment and removed certain transcription factors. And we got it down to a 3-factor combination, ETV2, SOC7, and NR2F2. And this would give you ectopic vessels at a very high level. Now there's some redundancy in the system. ETS1 will work for ETV2. And more recently, we've shown SOX18 will work for SOX7. But NRF2 seems to be unique. So if you inactivate NRF2 with a morpholino and antisense type of approach, you lose the expression of this transgene. So the NRF2 is required for the expression. And we also show here, if you're looking at red stem cells, that if you give the NRF2 morpholino, you reduce the number of stem cells in the niche. So these stem cells actually bypass the niche and don't land as much. So I think this is very good evidence of the function of our particular code. So then Elliot Hagedorn, the very talented postdoc who did all this work, came to me one day and he said, you know, this is the best day I've ever had as a graduate student or a postdoc. And I said, well, what's going on? And he said, well, I made an ectopic niche in the background of a fish that has red stem cells. And in this ectopic niche, the stem cells are recruited there. So then I got very excited and I said, well, you know, do the niche cells cuddle the stem cell? And he said, yep, they do. You can see this here, cuddling the stem cell. And so then I asked kind of the home run result, which is, do they divide in this new niche? So this is a movie. And what you'll be able to see is this is the normal niche down here at the bottom of this screen. And you can see a lot of stem cells in that niche. Now, by chance, in this one animal, we were able to capture an ectopic niche that's rather small up near the top of the embryo, near the spinal cord. And you can see that it's already recruited some ectopic cells. So this niche seems to be interesting. But now I'm going to play the movie, and if you could watch this upper niche, what you'll see is those stem cells in that niche are dividing. So we feel that this is a demonstration of fully reconstructing a hematopoietic niche by reprogramming technology. Now we've played with a number of different promoters to see what cell types can actually be use the reprogramming factors to make a niche endothelial cell. Muscle can be turned into these cells. Certainly endothelial cells can be turned into these cells, and you can see that very nicely here. But there are some tissues that can't be turned into these cells. So we're working on how broad this type of approach would allow a reprogramming. Now, the other thing is the work that I was showing you has to do with a fetal niche or a larval niche. And we wondered what about these factors in the adult marrow. And what I can say based on single cell RNA-seq of adult marrow, clearly these factors are expressed. So then we collaborated with Shaheen Rafi's lab. And Shaheen has done a very nice experiment through mouse development, dissecting out a variety of different tissues over time. And then we looked at our signature, and what you can see is that at E14 to E15, you see the fetal liver has very high levels of our factors. This persists to E16 and E17. Remember, the fetal liver is the site of hematopoiesis, but at birth, it switches to the bone marrow. And you can see it kind of goes down at P2 to P4. And then in adulthood, what you can see is the venous sinusoids of the marrow express a fair amount of our factors. So again, we think this is evolutionarily conserved, and the niche endothelial cells in your marrow are the same as the niche endothelial cells, let's say, during fetal life. So in this story, we've used RNA tomography. We found 29 genes that are very specifically expressed in the niche endothelial cells, the venous sinusoids. We found a code, and then we used factors to reprogram ectopic niches. And we're in the process right now of taking human iPS cells and reprogramming them to these niche endothelial cells. And the idea might be in a patient who had myelofibrosis, you might be able to inject these cells into a new site, let's say in somebody's arm. And then the arm would become a marrow. And that would obviously rescue the stem cells that are being pushed out of the marrow, and they would have a place to divide. So those are some of the concepts that we're thinking about. Now I'd like to transition to the talking about MDS and AML, particularly in clonal hematopoiesis. So what happens in normal life is you're born with a number of hematopoietic stem cells, and these are polyclonal. But a number of years ago, some investigators, including lab of Ben Ebert and many others, I should say, started sequencing patients who are old, not even patients, just normal humans who are old. And they found that there are mutations that occur in epigenetic regulators and splicing factors and other things that are involved in clonal selection. And these clones expand and can often get up to 20 to 30% of the blood is being driven by a small number of stem cells. Now, obviously, this will predispose you to having myelodysplasia and ultimately leukemia. And the question is, how are clones balanced? Is there a way of reversing this type of a process? And what is the impact to leukemia? Now, if you think about aging and mutations, if you would have asked me about 8 years ago, how do people age, I would have said it's epigenetic. But recently, based on the chip here, the blood mutations, a number of other investigators have started to look at mutations in other tissues. And there's lots of mutations. So for instance, a plastic surgeon took off eyelids of elderly women and found clonal mutations in P53. Some esophageal biopsies from aged individuals showed large clones of Notch mutations. And very recently, there's some interesting data, and Matt Meyerson talked about it also recently, about expanding clones in the intestine. And so what we may find is actually aging ends up being mutational, and this clonal selection is quite important. to study. And I think that, the blood system is actually one of the perfect systems to be able to study this. So we wanted to look at different clonal studies, and one concept that we had was to actually label every stem cell in a different color. So what we did here is we started with this fish called the rainbow fish. This has red, blue, and green separated by LOX P sites. When you mate this to a blood-specific CRE-ERT2, in every cell type, you'll start getting random inactivations of these colors. And this gives you shades of colors, similar to the old RGB TV sets that many people may remember. So you can give tamoxifen at a certain time during development, label every single blood cell a different color, and then basically grow the animal up to adulthood and look at the colors that happen in adulthood. Now remember, I told you that the birth of the blood stem cells is happening roughly around 24 to 30 hours in a fish. So the number of colors that you would get in the blood would loosely resemble the number of stem cells that are actually born during embryogenesis. So we did this, and this is a picture of a fish that has multicolored blood. And what you can see is all the blood is different colors. And it's really, really pretty in person. But what you can see here is this one cell that's a blue-green cell in the middle in one of those holes. Based on the position, this is very likely to be a hematopoietic stem cell. Now you can see it dividing. And when that cell grows up to adulthood, you'll make blue blood. So we then went ahead and grew the animal up for 4 months, and we did a peripheral blood smear. And this is the peripheral blood smear. So I'm a hematologist, and I can tell you that's the prettiest blood smear I've ever seen. Basically, you can back calculate the colors now. And the important line here is that 24 hours of development, when we've given tamoxifen, there are 21 stem cells made in the aorta. And if you give it later, you increase the numbers, but basically, that's the number that starts off the entire hematopoietic program. So this now allows us to mark clones and has set us up to do some really elegant experiments. So first of all, remember in polyclonal hematopoiesis, if an aged individual acquires mutations, then you get clonal expansion. But in our case, we're going to label every stem cell a different color. We can put in CRISPR or put in a transgene and then watch clonal expansion happen in these fish. So this is the work of Serena Vagian, and she's created this technique called Twister, which is tissue editing with inducible stem cell tagging via recombination. Many of you have probably played the Twister game with the different colors, so hopefully you appreciate the analogy. So what Serena has done is to inject 20 guide RNAs, which represent the mutations that occur in humans. We've also included pseudogenes to have controls, and then we inject Cas9 mRNA. It's very important for us to inject the mRNA because it's not as potent as the protein, and this will allow mosaic mutagenesis to happen. So what we're doing is in vivo competing a normal stem cell against a mutated stem cell and seeing who wins. We then give our tamoxifen to give the fish the colors, and then we can look for clonal expansion later and study those clones. So this worked incredibly well. As you can see in the control, very few fish have any dominance, and probably these fish that do have a little bit of dominance, it's where the colors are too close for us to actually tell the difference. But clearly putting in the pool, you get fish that are very, very have dominant clones. And you can see like this fish up here has 70% of the blood is coming from a single hematopoietic clone. So what's interesting about this is all of the mutations that we see in the fish are exactly what you would see in humans. So if you look here in terms of the number of fish with mutations, we got ASXL1 and P53 and a DNMT3 ortholog, PIG-A and a STAG2. So mutations are very, very similar. I think the average fish has like 5 or 6 mutations, and we can verify all these things by sequencing. Now, there are some alleles down here that must be required for early embryogenesis in some way. And so these we're not actually getting enough clones to make conclusions about. Now, the advantage of labeling a stem cell in a different color is for the first time, you can actually sort out the stem cells or sort out the blood and start to evaluate, what's going on in those cells to make them dominant. This allows us to compare a dominant versus a non-dominant clone in the same fish. Now, we know these things are clonal not only because of the color, but we can also sort each color and sequence them to see what mutations they have. And that also demonstrates clonality. So multiple QCs for us to show that these are clonal events. So then we compare dominant clusters versus non-dominant clusters. And what we see is ASXL1 clearly selects dominant clusters. And I just want to point out, this is a mutation in exon 12. If we knock out the entire gene, you do not get a dominant clone. You must do exon 12, and this is exactly what we see in humans. And then you can look at the different mutations, and we do see that many of the mutations are present in dominant and non-dominant clusters. So it's very interesting, and what we've been able to do then is to do single cell RNA-seq on the dominant cluster versus the non-dominant cluster. And what we've been able to find is similar to other investigators' results. The macrophages certainly pump out a lot of IL-1 and TNF-alpha. And this has been linked to endothelial damage and also cardiovascular risk. But we also see that the stem cells have suppressors of inflammation. And this is quite interesting because we're able in the same animal to look at the macrophages and the stem cells. And it presents a model, which Serena Vagian, the postdoc who's done all this work, has done, is that, perhaps what happens is these macrophages can start making things like IL-1 and TNF-alpha, but then the stem cells respond to the inflammatory milieu by actually increasing inflammatory modulators, things that counter this response. And that may allow for actually the selection. So the other thing we thought we would check with this system is to look at kids who have congenital defects that cause increased in clonality. This is true of patients, let's say, that have GATA2 mutations or RUNX1 mutations who have a variety of familial disorders that can lead to clonal dominance. So what we did is we used our rainbow system in the background of the RUNX mutants in this particular case, and then collected and assayed clonality. So what we can see here is, I should say, our RUNX knockouts actually live to adulthood. In the zebrafish, there must be another pathway that actually helps, because in a mouse, the RUNX knockout is dead. But you can see clearly the cluster size as well as the average skewing is certainly affected here. And so what this indicates is that a deficit of hematopoietic stem cells early during life can also lead to a dominance. So said by a model here, in the acquired state, let's say typical aging, you might end up getting clonal hematopoiesis as a result of a mutation shown here with the green. And then subsequent mutations lead to an expansion. But here in the germline state, where you're actually a RUNX1 mutant, what may happen here is you end up with oligoclonal hematopoiesis. Thus, you are clonally dominant, but the reason for being dominant is you have less stem cells from these clones compared to the case here where actually you have an expansion. And then, interestingly, the malignancy state looks very, very similar between aging and these young kids because of these secondary mutations. So I just want to fill you in on some of our newer studies, which we're really excited about on barcoding stem cells. And this is the work of Sarah Rubin and Chloe Barron in the lab. This Gestalt fish actually won the Breakthrough of the Year in Science magazine in 2018. And what happens in this situation is you can have the ability to inducibly heat shock and essentially using CRISPR barcode every cell with a different DNA sequence. Now, what happens here is you have a stage 1 editing in the fish, and we can inject in, let's say, Cas9, and basically barcode the first 4 positions with guides 1 to 4. But then we can later heat shock this and have the Cas9 being expressed, and then barcode positions 5 to 7 with different guide RNAs. So again, this allows for a broader view than the colors in terms of actual barcodes, but it's been quite successful in the lab. I do want to say that the Gestalt system has gone through several different versions, and we're kind of up to version 3.0 at this point. So a lot of technologic advances to be able to do this. So this just shows an example of one fish, and we looked at different clones in the fish. And again, this is based on single cell technology, single cell RNA-seq, and you read the barcode from the RNA. And so you also get the lineage of the cell as well as the barcode. And that's one of the advantages. So here's an unbiased clone. In other words, you can see the B cells, the T cells, and the granulocytes, as well as the stem and progenitor cells have clones. I should say have cells that are participating from this clone. Then here's a lymphoid-specific clone, okay? And here is a myeloid-specific clone. So this technology certainly seems to be working well. And one of the things that we found, which is quite surprising, is a fair amount of the adult situation is that there are stem cell clones that are only lymphoid specific. And these are obviously long-lasting clones because we wait for months until the fish is really an adult to test the clonality. So I think we'll be hearing more and more about the use of Gestalt in this particular process, and another exciting development on cellular barcoding. So I want to kind of finish up with a pretty significant story on macrophages and clonality. And this is the work of Sam Watrous in the lab. So Elliot Hagedorn had done some imaging studies which were quite interesting, looking at the interaction of macrophages with stem cells. And here you have red macrophages and green stem cells. And what you can see is actually the red macrophages is actually grooming the stem cell. There's an intense interaction here, and you can even see that chunks of cytoplasm are being removed by that macrophage. I'll just play it one more time, just so that you can see it. But you know, you can see the green material being deposited in these macrophages. And so we wondered what the functional consequence of this macrophage interaction would be. So Sam Watrous then started doing more imaging. And I'm going to show this video, and there's a couple different cellular behaviors I want you to see. So the first thing that happened was a surprising thing that over here on the right, we saw about 20% of the stem cells that are born end up going to the next niche, and they are eaten and become dead. So that's a surprise, you know, that the animal would go to the trouble of making stem cells and then several, 24 hours later, be eaten by macrophages. But then we saw another behavior where basically the stem cells are interacting with the macrophages. And these cells, when they get groomed, they end up dividing. And so basically you see that stem cell is about to divide right there. And it's pretty surprising. It's almost as if the macrophage stem cell interaction is a licensing event for the stem cell to divide. Now, it turns out about 20% of the interactions in this niche are ones where the stem cell is killed. And 80% of the time, the stem cell will be partially eaten, and then that stem cell will actually divide. So obviously, these imaging interactions make you have a lot of questions, right? What's the frequency of these events? What determines this kind of interaction? Is there anything special about these macrophages? And, what determines a doomed cell versus a groomed cell? And I think another central question is, what's being monitored on the stem cells and why does this even happen? So one thing we can say is about 20 to 25% of the RUNX positive cells are engaged with a macrophage in the niche at any time. And we were able to look over time, and this number is relatively stable throughout all of development. So, I think it's a natural process by which macrophages are looking at stem cells. Now, we were also able to look at specificity, and what we can see is that the macrophages are interacting with the stem cells more specifically than they ever interact with red blood cells. So again, the interactions we're seeing appear to be cell specific. So within the zebrafish toolbox, it's possible to delete the macrophages. And so we can give a drug like clodronate liposomes, and they are taken up by the macrophage, and this kills the macrophage. And you can see that this animal has very, very few macrophages. Now, we also have the ability to use an IRF8 morpholino. And this interferon pathway is responsible for the macrophage lineage. And you can see this tremendously reduces the number of macrophages. So one question that we had initially is if you delete the macrophages with these different techniques, then what would be the effect on the RUNX positive cells? And what we can say is basically there doesn't seem to be a change in the absolute number of RUNX cells per embryo. And you can see this either for the IRF8 morpholino or for the clodronate treatment at 2 different time periods. So then we have this new technique on clonality. So we decided to assess what happens to the stem cell clones if you deplete the macrophages. And you can see here at 24 hours, we would give the tamoxifen, labeling the stem cells in different colors. At 28 hours, and I'll show you subsequently, we will add clodronate, depleting the macrophages, and then look at the stem cell number several months later. So the surprise here is if you look at the clones, the clodronate animals have half the number of colors compared to the sibling controls. So this is a substantial reduction in clonality. Now, what we did is we initiated our studies at 28 hours giving the clodronate. And you can see here that basically the sibling control has about 25 clones, and the clodronate has about 15 clones. This is in adulthood. When we look at an IRF8 morpholino, we see a very, very similar approach. And then because the clodronate given at 28 hours might be close to when the stem cells are being born, we decided to give clodronate much later at the point when they're arriving in the niche. And you can see again, there's a very impressive change in clonality, a reduction in clonality. So, what's going on here to account for this kind of step? And we obviously think there's an intimate interaction between the macrophage and the stem cells. And this is affecting the division of those stem cells to affect this clonality response. So the macrophages have actually eaten some of the stem cell, and it's possible to use flow cytometry to sort out the macrophages that have red inside them, having eaten a red stem cell. And this is on average about 0.4% of all the macrophages in the animal, but it allows us to characterize these cells really in depth. So the first thing is that the macrophages express genes that are important for engulfment. So you can see a number of pathways represented. This is, again, single-cell RNA-seq or bulk RNA-seq, but you can see these are complement, and you can see S1P receptors, and these are certainly involved in migration pathways and other things. one of the other very interesting things was there were a number of apoproteins that are involved in cholesterol transport that were very high in the grooming macrophages. And so we wanted to investigate this a little bit more. And Sam was able to find a cholesterol Bodipi dye, which is a dye that can be used, and you can then watch the uptake of this dye by macrophages that might be interested in the cholesterol. And again, this works very well in the zebrafish system. And what you can see here is a macrophage, and you can see that it's RUNX positive because this RUNX positive cell is abutting the macrophage. And then you can see the Bodipi dye is coming into that particular macrophage. So again, the macrophage has these pieces of RUNX in them after it had arrived, and those are co-localizing with this Bodipi cholesterol dye. I should also say that ultimately you can see these spotted pictures in these macrophages, and those spots end up being lysosomes. And Sam has been able to also trace these with a lysosome marker. So when we look, the macrophages that are degrading the RUNX M. cherry population also are looking, they are taking up the Bodipi dye. And this is, again, almost 100% of them are. So we wanted to study this a little bit more at the single cell level. And we decided to do an index sorting approach called SortSeq that actually Chloe Barron in the lab had worked on when she was a PhD student. What you do is you can sequence the cells in each well as a single cell. So the macrophages really nicely came out into bins here, and you have this kind of progenitor-like state. And although I'm not 100% comfortable saying M1 or M2, the genes that we see up here are a little bit more M1-like than this M2 population. And we think that it's this particular population in here that's probably the one that's driving this grooming behavior. So when we look here, you can see that NF-kappa B signaling, IL-1 signaling, reduced interferon pathway, and then the lipid metabolism is present in this particular process, as well as some endothelial signaling. So this makes a lot of sense for us. And we wanted to be a little bit more precise. The nice part about index sorting them and having the FACS profile is that you can basically go back and look at the IDs and then overlay them and see where the patterns actually came from. And of course, you can see that these populations often are intermixed, but now that you know the actual result, you can, in transcriptional space, separate those. And that really is helpful to understand what genes are being expressed. So when we do this, we see a very strong lysosomal compartment by gene expression. And as I showed you, the cholesterol is taking up into the lysosomes. And we have genes like HMOX1, which are really good markers of these specific macrophages. Now, this is a gene that's a heme oxidase gene that's typically used in the spleen to break up red blood cells, but it obviously has an oxidation pathway. And so it might also indicate some of the state of the oxidation that's actually going on. So another question that came up was, if a stem cell is coming in and it's going to get attacked by a macrophage, is that stem cell kind of already dead? Or is it a perfectly happy stem cell and just gets ready to interact with the macrophage? So in this particular study, we used acridine orange. And what we can show is that the RUNX1 positive cells that's coming in here is not acridine orange positive, but once it gets chewed by the macrophage, then it's acridine orange positive. So again, this indicates that the stem cells that are born in the aorta seem, relatively healthy before they get eaten by these macrophages. Now, we also were able to take the hematopoietic stem cells and progenitor cells and do RNA-seq with those. And what we were able to show is there's really no transcriptional difference in the stem cells that have been eaten. And, we think, therefore, it's probably a biochemical pathway that's actually driving this process, like a signaling pathway or something like that, or perhaps some type of code on the stem cells that's being interacted with the macrophages. So more recently, we've begun to do whole cell proteomics, and a group at Harvard has perfected proteomics on as few cells as 120 cells. And this allows us to sort these cells and see what are the factors that are present in the stem cells or in the macrophages or the macrophages that are eating the stem cells. So one of the factors that was differentially expressed by the eating macrophages was calreticulin. And these other factors are also differentially present by protein work. And we see CalR and CalR3A and 3B. These are evolutionarily conserved calreticulins. From our knowledge, there's no functional difference between calreticulin 3 and calreticulin R, but nevertheless, this has seemed to come up in our study. And this might be the type of signal that could be an eat-me signal for these stem cells. This is based on work of Irv Weissman and others that have shown factors like CD47 are eat-me signals, but also calreticulin is very, very important. Now, the fish does not have a CD47 ortholog or a SERP alpha ortholog, so we don't think that system is what's being recognized here. But if you think about calreticulin, what we know is that calreticulin is typically an ER protein, but it has a KDEL sequence, and that KDEL sequence can be cleaved. This allows calreticulin to then get transported to the surface, the plasma membrane surface, where it can actually bind to individual proteins and be presented. There's actually a receptor, as I'll talk about soon, called LRP1 that would be a receptor for this kind of presentation. Again, through the work of Irv Weissman and others, you have these recognition models where you have SERP alpha and you have CD47, but it's also possible where you'd have calreticulin on the surface being recognized by LRP1 on the macrophage. And I should also point, it's possible that the macrophage could even be the source of the calreticulin, secreting it onto the surface of the stem cell, and then this interaction can also happen. So we were lucky enough to have a calreticulin antibody that works in the zebrafish. And these are RUNX positive cells that have been sorted. And we see calreticulin on the surface of the stem cells. And when we look at the macrophages, we can see that the receptor is present for the macrophages. So this would allow this interaction to occur. So what Sam has done is to inactivate calreticulin and calreticulin 3 orthologs and look at the interaction between the calreticulin and the stem cells. And what we see is a reduction, particularly with calreticulin 3, of the number of stem cell macrophage interactions. And Sam has actually done this in the background of the rainbow fish. And those fish are growing up, and if we ever can get back to the lab, we will be able to assess the clonality that's involved. So just to finish up, our model is basically that when you're having this birth of blood stem cells, there's a lot of stress going on, inflammation and trafficking, division, and that allows 3 tracks to happen. In the first track, the stem cell never really interacts with the macrophage. That allows the stem cell to persist. We see a number of, like 80% of the time, where the stem cell will have the calreticulin. This will interact with the LRP1, and this could lead to the eating process, but this is a partial eating, and this is a licensing event for the cells to divide. Sam has used Fuchi to be able to look, and he sees initially some Fuchi interactions, which means that the cells are dividing after they have this macrophage interaction. Then another 20% of the time, the stem cell interaction is so intense, perhaps because there's high calreticulin on the surface of the stem cell, and that allows the stem cell to be completely eaten and removed from the pool. And this is maybe some quality assurance mechanism by which a niche is able to remove the stem cells that aren't functioning too well. You also might wonder, when a stem cell transplant happens, is how much calreticulin is on the surface and how those macrophages are interacting with those cells. So in terms of pathways, we think things like TNF-alpha and IL-1 have been linked to the birth of the blood stem cells. Maybe migration or shape changes could lead to the calreticulin being on the surface of those cells and interacting with those macrophages. And that would be some of the code that's being looked at for these interactions. So with that, I just want to thank the people in the lab who've done the work. I believe I mentioned everybody's name. And again, thank you very much. And I'm very happy to take questions.
Okay, thanks, Len. This is an amazing talk. I really enjoyed the movies and these beautiful pictures. So we have a long list of questions. We try to go through as many as we can live, and then the others could be answered online. Len can answer these. So there are a number of questions related to the first part of the talk, where you're talking about the endothelial cell niche, like specifically like why are those endothelial cells cuddle the stem cells? What activates this behavior? Is this related to peripheral nervous system? And does the cuddling influence the stem cell orientation of division? So these are a couple of questions combined into one. So maybe you can go.
Yeah, sure. That's great. Yeah. So, you know, what we know so far is there's obviously a very important endothelial hematopoietic stem cell interaction. And the interaction of the cuddling reaction, we don't know the mechanism. We were able to find a drug called lycorin that will actually affect the cuddling. And, but short of that drug, we think that processes like integrins are involved in this. And there's obviously some physical force necessary to actually be able to do the cuddling. With regard to the division steps and where they occur, it seems like division most likely requires the stromal interaction. And so, although the cuddling reaction is important to bring the stem cell adjacent to the stromal cell and to maintain kind of a structure, I think it's more this intimate interaction with the stromal cells that's driving the division step. So in that case, we had the TGF-beta pathway. If we inhibit TGF-beta, what you get are more divisions. And you can actually see a single stem cell divide once, and then if you've had it in the presence of a TGF-beta inhibitor, you divide again. And so this allows TGF-beta to be one of the pathways that's probably resulting in the niche being able to cause this division. We don't have any information about the nervous system at all. There are different neural cells that are present there. And so we're still investigating this niche, and there's just a lot of great questions to ask in the future.
Okay, thank you. So we have lots of questions on the macrophages as well. I will try to highlight, like pick some of them again. One question relates to the interaction of macrophages with the hematopoietic stem cells versus the erythroid lineage. Like, is there any kind of skewing depending on what is the eventual fate of those cells?
Yeah, so far that interaction can be reproduced in vitro or in vivo. And so basically the macrophages want to interact with the stem cells, and at least at this particular point when we're imaging, they don't want to interact with the red cells. So obviously there must be specific, codes. I think those red cells are very healthy. I don't think they're undergoing half-life issues or anything. So I think the interaction again becomes more specific, probably to this calreticulin response.
Okay, and then a number of questions related to like how the stem cells are chosen to be eaten, and you mentioned the calreticulin levels may influence that, but then also what makes the stem cells, the half-eaten stem cells, divide, and why the other ones don't do the same?
Yeah, so that's a really good question. So essentially, calreticulin is mutated in patients who have a myeloproliferative disorder, one of the labs working locally on this is Anne Mullally's lab. It's super interesting. And, this sets up a potential interaction perhaps into the TPO pathway or the JAK-STAT pathway. So you could have a situation where the macrophages are working on a receptor, let's say, and eating a receptor at the same time as they're eating everything else. And perhaps that's a licensing event to tickle these receptors and cause the division to occur. That's just one model among many, but I do find it attractive, if you had the TGF-beta receptor suppressing cell division and you were able to eat that partially, maybe you'd be able to divide something like TPO or KIT or something like that. A receptor type of process would be interesting.
Okay, and couple of questions again relate to why the stem cells would express core-reticulin and the other cells not, and if you ectopically overexpress core-reticulin in other cells, would those be eaten the same way?
Yeah, so I think it's pretty agnostic, and calreticulin is expressed by many, many, many cells. So any cell could actually do the secretion of the calreticulin. I think in Sam's model right now, we kind of think that the macrophage may be actually making some calreticulin and getting onto the surface of the stem cell. But it's also possible that the stem cell would make the calreticulin. I do think that the overexpression is a little bit tough because you have to put in a mutant that doesn't have the KDL sequence, because otherwise it'll get stuck in the endoplasmic reticulum. So we're doing those experiments now, specifically looking at the calreticulin levels and then seeing whether those cells are eaten. We're also overexpressing on our RUNX enhancer the mutated calreticulin that will get secreted in the stem cell, and seeing if we can drive the process so more stem cells are actually eaten.
Okay, then questions related to the macrophages, like are different types of macrophages are different ability to eat the cells? I think you kind of mentioned the M1, M2 difference.
Right. I think pretty much we feel that these are a subset of macrophages. They express that heme oxygenase. So, I think it's a cell that's poised to be looking for another cell so that it can clear that cell out. And so I think that interaction is specific.
Yes, and the question also related to this is like, is this interaction conserved in mammals? And my personal question, because in the mammary gland also macrophages seem to play a role in being a niche. So do you think this could be like a general phenomenon in other types of stem cell niches as well?
Yeah, so it's very, very possible. We have not investigated the other niches or in other tissues, but I think when you have a group of macrophages being present, it's possible that certain populations of cells are going to be culled out, and this could be done by this calreticulin, and I think as Irv has shown could be CD47, and these types of mechanisms of eat me and don't eat me signals are quite important, as the niche is developing, and then also as the niche is functioning. And I don't know, for instance, in the hematopoietic system, whether this is only operative during colonization. And that may be my suspicion, is that the stem cells function probably pretty well if they've engrafted. But maybe this is a system to basically watch out on engraftment of stem cells that may not be as healthy. And so I think it's kind of a quality assurance mechanism.
Yes. So we have time, I think, one more question that's related to core-reticulin mutations that are found in subset of patients with myeloproliferative neoplasms. Do you know if those mutations would affect this macrophage eating of those stem cells? And also, do you know if in patients, again, if the lipid protein levels, the lipid levels in the blood would influence this interaction?
Yeah, very, very good question. So, sorry, do the first question again, Nelly, just to...
Yeah, the core-reticulin mutations, would they influence the...
so I think so based on the work, the genetics would suggest that the calreticulin is really going through a TPO pathway, JAK-STAT pathway. So we don't think this is necessarily going to cause the same effect of being eaten. I do think if we put these factors into our fish, we'd get a myeloproliferative syndrome. And so I think that, ultimately would happen. And then on the second question, Nellie, sorry.
That was related to the lipid metabolism. So
Yeah.
patients who have, perturbations of the lipid levels or lipid metabolism, would that influence this interaction?
Yeah, I think it easily could. I mean, obviously, there's a competition of the macrophages eating those cells, which is going to involve cholesterol. When people have looked in the mouse at looking at TET2 mutations that causes clonal hematopoiesis, if you've given them a high-fat diet or given them an LDL receptor knockout on top of that, you end up getting severe vascular problems. And clotting and, the cardiovascular risk is substantial in these patients who have clonal hematopoiesis. So I think, given people who have lipid issues or the local lipids in the niche could easily be involved in this kind of, in modulating this effect that we're seeing.
Okay, thank you very much. We have additional questions, but we don't have time to answer these online, on live, but again, like Len will have access to those questions, and many of them are very good. Thanks for being so active. And I thank again Len for the wonderful talk. And also, just as a reminder, the slide presentation will be uploaded on the website. You can look at it later on. And also, Len will try to answer the questions online, and we'll appreciate that. So we continue this series. Next week, we have an amazing lineup of speakers, and hopefully many of you will be able to join next week as well. Thank you very much. Have a nice weekend, everyone. Bye.
Thank you.